Hello all. We'll wait for 5 more minutes to let others join and then we can start. Hello all. I'll just share my screen. Can you people see the slideshow mode? Anyone can let me know if you can see this?
Yes, ma'am.
Okay, yeah, thank you. So, good evening, everyone. Welcome back to week 6 of PMRF TA sessions for the NPTEL online course Drug Delivery Principles and Engineering. As you all know, we have been discussing few concepts of drug delivery and tissue engineering, and are also discussing some of the previous year assignments, which will then help you for your final exam. Yeah, so I'll just start with some concepts which were discussed in this week. So you must have already been through some of the lectures for this week. So as you all know, we have discussed quite a lot about drug delivery, various methods of drug delivery and various techniques. And in the last lecture, we got introduced to something known as tissue engineering. So what is tissue engineering exactly? It's a mix of using various principles of engineering and cells and biomaterials to replace or to revive a damaged tissue in a human body. So it could be to generate a whole tissue in vitro and then replace it in the body, or it could be you can regenerate a part of tissue that is damaged in vitro, like outside the body. In vitro means outside the body in the lab, and then replace the damaged portion. Or you can put scaffolds to regenerate that part of the tissue in vivo, or it could be something known as adding support material to enhance the function of any damaged tissue. For example, you have bone implants in case there is a fracture. So this was about tissue engineering. Then next, since our course is drug delivery, we have to look at why drug delivery becomes important in tissue engineering. Yeah, so as I was saying, first of all, why do we need drug delivery in case of tissue engineering is because there are various instances where we might need certain small molecules to be delivered along with the organ that you are implanting. For example, let's say you have made an artificial liver and you're transplanting it into the patient's body. So this artificial liver, whatever you have made, is going to be a very thick tissue. And once you implant it, the cells which are present inside the liver, your artificially made liver, they should also be getting nutrients as well as oxygen. So now if it's a very thick tissue, your oxygen and your nutrients and the waste that is generated by the cells, it won't be able to reach outside. So that's why it needs a lot of blood vessel formation. So to enhance that blood vessel formation, there are certain growth factors for the same. So along with your tissue that you have implanted, you also need to deliver these growth factors. Or it could be that you need antibiotics to be delivered along with the tissue to avoid infection, because now you have made something in the lab, you have made an artificial organ and you are implanting it into the patient's body, and during the surgery and all, there are a lot of chances of getting bacterial or fungal infections. So to avoid that, you also need to deliver antibiotics along with tissue engineering. Or there could be immunosuppressants. For example, you have identified a donor and you have decided to do a surgery and transplanted that organ in a different patient's body. Now in that case, since the patient's body is going to consider this transplanted organ as foreign, it is going to launch an immune attack. So we don't want that. We don't want the patient's body to degrade the transplanted organ. So in order to do that, you have to suppress the immunity. So there are several immunosuppressants present which you have to then deliver along with your transplanted tissues. These are some of the examples where you need drug delivery concepts. So one such example of delivering molecules is growth factor delivery, as I was talking about for blood vessel formation. So there's one way where you can just deliver the growth factors, but growth factors are very small molecules, so they have very less retention time in the body. So once you deliver them, they are going to be quickly cleared out of the body. So we don't want that. We want the growth factors to sustain in the body for a longer time, so that it helps in the regeneration of tissue. So growth factors are nothing but these are small molecules. These could be proteins. They are involved in cell proliferation. They are signaling molecules which then help in differentiation of cells from the stem cells to a particular type of cell. So that's why growth factors are very important when you're looking for regeneration of any organ. So this you can do. So as I said, if you just deliver the growth factors, there's a chance that these will be cleared out from the body very soon. So to avoid that, you can deliver growth factors along with biomaterial. So we have been looking at a lot of these drug delivery techniques like a reservoir system or a matrix system. So something like that you can do with the growth factors also. So to do that, first of all, what you can do is you can select biomaterials in such a way that they will have a natural affinity for this growth factor. So your growth factor can directly go and bind to these biomaterials, and then you can deliver these growth factors inside the patient's body. Or in case, let's say you're not able to identify a particular biomaterial and the affinity for this growth factor to the biomaterial is very less. So in that case, what you can do, you can use heparin. Heparin is one glycosaminoglycan glycan. So it's a polysaccharide which is naturally present in a human's body. It's a component of the extracellular matrix, and heparin has a lot of binding sites for various different proteins and ECM components. So what you can do, you can take your biomaterial, it could be any polymer which is compatible with the body and the growth factor. So you can take that biomaterial, you attach heparin to it, and then you can then conjugate your growth factor to it. So it's going to be a system wherein you have a polymer chain to which your heparin is attached and to which your growth factor is attached. So this is one method of delivering growth factors for tissue engineering. Or you could use ECM fragments to further enhance the affinity. These are all non-covalent methods of delivering growth factor. You could also use covalent cross-linking. For example, you can use a particular chemical reaction to conjugate your biomaterial to the growth factor. So this was one of the concepts discussed in this week's lecture. Then, so this was about drug delivery and tissue engineering. Another important concept that was discussed for this week is implant-associated infections. As I said, since these implants, let it be an artificial organ, or let's say you have a bone implant and you have implanted it into a patient's body. Now during this, there are chances that so first of all, it could be possible that your implant was not sterile when you were implanting it. It could already be carrying some bacterial infection, or during the surgery there might be the environment is not clean, or the tools that were used during the surgery were not clean, and somehow bacterial infection gets introduced into the patient's body. Now this is something known as implant associated infections. The bacteria that is present in the implant, it establishes itself into the patient's body and leads to further infection. So this is a very big problem in tissue engineering and drug delivery. So there are a number of ways to avoid that. So in this lecture, there are two important concepts related to implant associated infections. One is biofouling. Biofouling is nothing but it is attachment of, let's say, proteins or cells or bacteria to a particular surface. So it forms a layer all over the surface of the implant. And another concept is biofilms. So biofilms is nothing but the microbial colonization. So let's say you have a surface and your bacteria gets attracted to the surface. Slowly it will establish itself on the surface of this particular implant, and it is going to form something known as a biofilm. Biofilm is a layer of these bacteria. They kind of colonize on it. They form their own colonies there, and they form a very thick layer which could then include polysaccharides. They could include various components. So in a way it becomes very dangerous now, because since the bacteria is now living there, your antibiotics and your immunity of the body is helpless against these biofilms. We'll discuss more about this in the upcoming questions as well. So biofilms is a very is a very dangerous thing and is quite a problem for implant associated infections. So what happens exactly is, when you implant a particular material in the patient's body, there's all of the things will try to interact with the implant. There could be proteins in the body that are interacting with it. We saw protein adherence and protein attraction of proteins towards the implanted surface in the previous lectures. So in the same way, cells of the patient's body will also try to interact with the implant as well as the bacteria. So so there's kind of a race happening in between all of these things of who is going to reach the surface first and who is going to establish themselves on the surface. So in this case, the bacteria wins the race, because bacteria evolutionary has been adapted to colonize on surfaces, and it's also present in large number. So the bacteria will rush towards the implant, it will adhere and colonize over it, and as I said, it will form a biofilm. Now your immune system, for the immune system, the bacteria is still foreign, so the immune system will try attacking this bacteria in order to kill them, but they are not able to do it. So lots of inflammation starts happening around the implanted device, because your immune system is attacking it and it's not having any effect on the bacteria. So in a way, the implant that you have put into the patient's body is leading to more and more problems. So the patient starts getting a lot of problems because of this infection and the immune attack. So in a way, the only solution to this problem is then you have to remove that device. So in that case, this therapy fails. So that's why this concept becomes important, where you have to understand how implant associated infections happen and what are various ways to overcome that. So some of the ways were also discussed in this week. These could be, you have to make a very careful choice of biomaterial. For example, there are few biomaterials, for example polymers, which don't have very good affinity for bacteria. So rather than, let's say you have a choice in between polymer A and polymer B, and you know priorly that polymer A is very good for bacterial colonization, versus polymer B is not good for bacteria. So bacteria does not get attracted to polymer B. So if you priorly have this knowledge, then you can make a careful choice of biomaterial, that no, I'll use polymer B for my particular implant, and in that way you can minimize the interaction of bacteria with your implant. Or you could coat with antibiotics in order to avoid colonization of these bacteria. Or you could make the surface more hydrophilic, because bacteria, they like hydrophobic surfaces more. They try getting attaching themselves to hydrophobic surfaces. So in that case, if you make your implant surface more hydrophilic, this could be one way to minimize the bacterial interaction with your implant. So these were some of the concepts that were discussed in this week's lectures. And with this knowledge, then we can go ahead with the we can start discussing some of the questions of previous assignment. Before that, if anybody has a problem with these concepts, we can discuss that first. Anyone has any doubt, or should I move on towards the questions? Okay, then I'll start with the questions. So I hope you can see the question displayed on the screen. So the first question is, which one of the following is incorrect about biofilms on implant surfaces? And we have few options here. So take your time in reading through the options. If anybody wants to volunteer, they can. And after a few minutes, we'll discuss the answer. Anyone would like to volunteer? I hope you guys can hear me. I'm audible, right?
Yes, ma'am. Yes.
Okay. Okay, somebody is trying to answer in chat, so he's chosen third option, it increases antibiotic interaction. Okay, we have one answer. Anybody else has a different answer? No? Okay, then I'll discuss the answer. So yeah, you were correct, whoever answered in the chats. The correct answer is, it increases antibiotic interaction. So read the question carefully. It's asking us which one of the following is incorrect about biofilms, and we were discussing about biofilms right now. So this is what biofilms does to the bacteria. So it essentially inhibits the host defense mechanism by making bacteria more powerful. So how it can do that is by various different ways. It will prevent the antibody penetration to bacterial surface. For example, now let's say you have free floating bacteria around and you have antibodies in the patient's body. Antibodies are very powerful, they're very specific, they can easily identify the roaming bacteria, and they can act against the bacteria. Now instead of that, if you have a layer of bacteria, very thick layer of bacteria, that will give a kind of protection to the bacteria living inside that biofilm, because antibodies won't be able to penetrate to the bacterial surface. So in this way, it inhibits the host defense mechanism. Next could be, it prevents macrophage bacteria interaction. So macrophages, you might be knowing, are the most important phagocytic cells in a human body. They are most important cells of immune system. So macrophages are able to directly engulf the bacteria and kill it. So biofilms will prevent the macrophage bacteria interaction also, and they'll also reduce the leukocyte interaction. It could be leukocytes are white blood cells, so it also reduces the neutrophil interaction with the bacteria, again making it more powerful. Or it could enhance virulence of the bacteria. For example, let's say in the biofilm, various different kinds of bacteria are there, and let's say initially there is a bacteria which is not virulent, which is not harmful for the patient's body. but it is now living with another bacteria. So it is possible that because of the presence of other virulent bacteria, the non-virulent bacteria can also turn virulent. It will also turn harmful. So it in a case it enhances the virulence of the bacteria because of the presence of biofilms. Or it could biofilm helps the bacteria in sequestering specific ions and nutrients, and it also prevents antibiotic interaction. So now your antibiotics are not able to are not accessible to the bacteria. So if they don't interact with the bacteria, they won't be able to kill the bacteria. So these are some of the advantages of biofilms for the bacteria from the bacterial perspective. So this question is asking us what is incorrect about biofilms. So the answer for this question is it increases antibiotic interaction, which is false, because presence of biofilm will reduce the antibiotic interaction. Is this clear? Am I making sense? Please let me know if I'm going too fast. I'll slow down, and if anybody has doubt in any question or if it's not clear, we can discuss again. Cool. Okay, I'll move on towards the next question then. So next is, why are stents used in blood vessels for tissue engineering? And you have to select two correct options. So be a little careful with these questions. Wherever you're supposed to select two options. So if I think last time there was a confusion in how the marks are given. I think if you select both the options are correct, you'll get full marks, and if either of it is wrong, you'll lose all the marks for that question. I think that's how they're grading it. So yeah, to talk about this question, go ahead with the options and let me know if anybody has an answer. Anybody wants to volunteer? No? Okay. Let's look at the answer. So the correct answer for this question is these two options: to open occlusions from vessels and to increase diameter of blocked blood vessel, hence increasing fluid flow. Let's look at what stents are actually. So stents are basically used to remove the plaques that are present in the arteries. So stents look like this. So it's essentially a metal tube which has a balloon inside, and so you must be first of all knowing that arteries do get blocked in older people. You must have heard people getting angiography, and then angiography is to know whether there are occlusions. So these plaques are also known as occlusions, whether people have that or not, and in case they have it, they do something known as angioplasty. So this is stents are used for that. So if you identify that some of the arteries in the patient's body are blocked, this could lead to heart attack if the plaque is not removed. So this plaque is nothing but debris which is accumulated in the arteries. So that's why it's obstructing the blood flow, and in that case then there's a lot of pressure on the heart to pump blood through these. So to remove this, what we can do is you take the stent. This is the metal, as I said, and it has a balloon inside it. So once you insert the stent in the artery, you blow up this balloon, you inflate the balloon. Now what will that balloon do? So because of the inflation of this balloon, the stent now becomes bigger. If you see in this image here, it essentially was of this size. Because of the inflation of that balloon, it has now become this size, and it kind of rubs against the plaque. It crushes the plaque, and then because of this, the occlusion or the plaque which is there, it starts dissolving, it starts degrading from here, and this way you can remove the occlusions which are present in the blood vessel. So that's where these stents are used. This is one kind of tissue engineering, you can say. So the question is, why are stents used in blood vessels for tissue engineering? So that's why it's used to open occlusions from the vessels, as we just discussed. And because of now that the occlusions are removed, it increases the diameter of the blocked blood vessel, which will then increase the fluid flow, the blood flow. Is this clear?
Yes.
Yeah. Thank you. Okay, then I'll move towards the next question. Next is, which of the following methods would you use to prevent bacterial adherence on device? We have to pick two correct options. I'll wait for you people to respond if anybody is interested.
hydrophilic surface and rough surface.
Okay, okay. Thanks for answering. So we have one answer where she's chosen rough surface and hydrophilic surface. Anybody else wants to give it a try? Okay, somebody's. Are you answering 2 and 3 or are you asking me 2 and 3? There's a question mark ahead of that. So let's say you're saying 2 and 3. Okay, anybody else? Thanks for answering. If there are no more responses, we'll discuss the answer. So the correct answer is option 2 and 3, regular smooth surface and hydrophilic surface. So as I said, there are various ways of reducing this bacterial interaction with the surface or the device. So there are different ways of doing that. So as I said, bacteria does not likes hydrophobic surfaces more. So in order to reduce the interaction of the bacteria to the device surface, you can make the surface more hydrophilic. So here there are various different, this is what I was discussing. If you have, there are particular biomaterials which favor bacterial adherence, there are few which do not favor bacterial adherence. So there are these ways of, you guys can go through this later on as well. And this is what I was talking about. Hydrophobic surfaces favor bacterial adherence more than hydrophilic, and also the shape of the device and the surface of the device matters. So let's say you have a smooth surface versus you have a rough surface. If you have a rough surface, it gives the bacteria more surface area to adhere to that surface. So in order to prevent bacterial adherence, you need to have a smooth surface, as smooth as you can make it, smooth surface, and then make the surface hydrophilic. So these are two ways of reducing bacterial adherence on device. Clear? Okay, then I'll move on towards the next question. Next is, which implant device has highest rate of infection? We have to pick one option. Anyone?
Option 5.
1, 2, 3, 4. Okay, heart assist devices. Okay, thanks for answering. Anybody else? Okay, we'll look at the answer. You're correct. Heart assist devices have the highest rate of infection. So this was discussed in one of the slides. You must have gone through all the lectures. So this just gives you an idea of, since we don't have any data from our country, this data is from US. So here are different devices, and then it's basically data telling us what different devices have, what is the percentage of the infections on these devices. So the most amount of infections you get are for heart assisted devices. This is also true for mortality, because heart is a very sensitive organ, and you can't really do anything if it starts getting infected. So that's where most of the infections happen, for device associated infections. Nothing much to discuss in this question actually, so I'll move towards the next. Next is, what are the common approaches used to reduce implant associated infections? We have to choose two correct options. Anybody wants to volunteer? We have been talking a lot about these concepts, so should be easy to answer.
Surface modification.
Okay, you have one option and you want to choose another. We have to choose two options. Thanks for answering. Oh, you're answering for this question, is it? Sahil 1 and 3. Okay.
Yep.
Sterilization.
Okay. Yeah, I think both of you are correct. The correct answer is sterilization and surface modification. So as we were talking about these implant associated infections, so the best way of preventing these implant associated infections is to sterilize whatever you are putting into the patient's body. So it might not be always possible to do so, but in case sterilization is not possible, you always have this option of surface modification. So these are just ways of, these are not like 100% foolproof methods of reducing implant associated infections, but these are some of the ways which you can minimize the infections in this case. So it's sterilization and surface modification. Clear? Okay, I'll take the next question then. Next is, which one of the following is not a feature of implant infection? So we have been discussing about infection. So read through the options. We have to select what is not a feature of implant infection. And let me know if you know the answer. Anybody? Any volunteers? Okay, 5 and 6. What is 5, 1, 2, 3? No, we have to choose only one option. So you have to either say 5 or 6. If you are answering for this question, of course. Okay,
Tissue
5 and 6. Okay, somebody is saying something. You're trying to answer? Okay. You want to try again? I think I lost your voice. Okay, cool. Yeah, I think you're correct. Tissue healing is the right answer. So, as we have been discussing about this, let's look at the options first. So, adhesive bacterial colonization on the material is what happens in case of implant infection. It leads to resistance to host defense mechanisms and antibiotics, which we saw in the previous answer as well, and it also can transform non-pathogens or opportunistic to virulent organisms because of presence of biomaterial substratum. Persistence of infection until removal of material, this is also a feature of implant infection. Absence of tissue integration, because now your implant is kind of covered with the bacterial biofilm, it does not lead to tissue integration. Last option is presence of tissue cell damage and necrosis happens because your immune system is now trying to attack the bacteria, but it's not able to kill the cells. But this, so persistent immune attack will lead to tissue cell damage and necrosis, killing of your own body cells as well. The only option that does not fit into the feature of implant infection is tissue healing. So implant infections don't lead to tissue healing. So that's why this is the correct answer. So you people can go through this later on. This is the same thing that we are talking about. So shall I take the next question if there are no doubts in this? Okay, so then next question is, how biofilms inhibit host defense mechanisms? We have to pick the false statement. Okay. Yeah, we just talked about this. So make sure you read the questions properly. You have to pick the false statement. Let me know if anybody wants to volunteer. No one? Okay, somebody is saying second option. Reduce virulence of bacteria and biofilms. Okay. Anyone else wants to give it a try? Thanks for answering in the chat box. No other response? Okay, let's discuss the answer. The correct answer is reducing virulence of bacteria and biofilms and making them susceptible to immune effectors. So you were correct, whoever answered in the chats. So for to answer this question, you first know how biofilms inhibit host defense mechanism. So this is what we were discussing in the last few slides as well. So preventing antibody penetration to the bacterial surface is one way of biofilms leading to host defense mechanism. Third option is preventing macrophage bacteria interactions is also true. Reducing polymorphonuclear leukocytes, chemotaxis is also true. Enhancing virulence of the bacteria, correct. Preventing antibiotic interaction is also correct. So all of these are methods methods by which biofilms will inhibit host defense mechanism. The only thing that doesn't fit into this is reducing virulence of the bacteria in biofilms and making them susceptible to immune effectors, which is false. Biofilms does not do any such thing. It does not reduce the virulence. If in case it just increases the virulence of the bacteria. So yeah, that's about this question. Any doubts? Okay, I'll take up the next question then. So the next question is, which of the following is an advantage of decellularized matrix? We have to select two correct options. Let me know if you people know the answer, or else we'll discuss. Anyone in the chats? No one? Okay, then we'll discuss the answer. So the correct answer is the matrix does not trigger immune response and the network of natural ECM is suitable for growing cells. These are the advantages of decellularized matrix. Let's look at what is decellularized matrix. So this concept comes from tissue engineering. So for example, let's say you have identified a donor who has donated an organ and you want to implant it into a patient's body to heal that particular patient. So since now these are two different individuals, there is a risk of strong immune reaction against the implanted organ. So to avoid that, we can use this technique. This is known as decellularizing the matrix. So what you can do, you can take that organ, for example, let's say it's a liver which you are transplanting. So to avoid the immune reaction, what you can do, you can remove all the cells that are present in this liver. So an organ will be made up of ECM as well as cells, right? So you remove all the cells. ECM is the extracellular matrix, which is actually the structure, the framework of this particular organ, and you just keep that. So ECM would be same in all the individuals. The cells you remove. So to remove that cells, you can use detergents. You can give it a harsh treatment which will kill all the cells, and then you can clear it off. Now you have this particular matrix system. So this is known as decellularizing the matrix because you have removed all the cells. Now you can take this matrix and you can put the cells from the patient's body into the matrix, they will then grow over the matrix because they have a structure. They have the necessary signals present in the ECM matrix for them to grow. So the cells will then colonize over that matrix and they will grow in that matrix, and they will then form the entire organ, and which then you can implant into the patient's body. So now this will what advantage will this decellularizing the matrix will give you? Since the matrix the organ is now made up of cells from the patient's own body, this will then reduce the risk of immune attack. So this is one advantage, which is here the matrix does not trigger immune response, and the network of natural ECM is suitable for growing cells because your cells have always been adapted to grow around the ECM matrix, which is natural for them. So if you do something like this, the cells will like the ECM and they will grow. Instead of having a completely different, completely artificial organ, you have made a natural framework from the ECM, which is then the native condition for the cells to grow, and they will then happily proliferate, and they will form the functioning of the organ will not be hampered. So these are two advantages of decellularizing the matrix. Is this concept clear? Okay, you were answering 3 and 4 for this question, I guess. I did not look at the chats. Thank you for responding. Any doubts here? No? Okay, let's move on towards the next question. I believe this is the last question for the assignment. The question is, which of the following is not an objective behind tissue engineering? Again, we have to select two options. Read through the options and let me know if you know the answer. Anybody? 3 and 6. 3, 4, 5, 6. Okay. Thank you for answering. Anybody else?
Option 3 and 6.
Option 3 and 6. Okay. Thank you for answering. Others want to give it a try? No? Okay, the correct answer is option 3 and 6. Both of you are correct. So we discussed about tissue engineering, right? Why tissue engineering is done. So it's done to generate new tissue to replace damaged tissue. It could be used to generate tissue in vitro, which can then be used to regenerate the damaged tissue inside the patient's body, or it could be to add support filler material to help the function of tissue, or putting scaffolds to help regenerate damaged tissue, which we saw in the decellularized matrix case. So the only two options that don't fit in this is embed cells like neutrophils and macrophages in scaffolds to enhance the inflammatory profile of damaged tissue. No, we don't want to do that in any case. Forget about tissue engineering. Why would you like to enhance inflammation is beyond my explanation, because this is the incorrect option. Or embedding cytokines like interleukin 1 beta and matrix metalloproteinase 13 to create more damage to the tissue. Again, we don't want to damage the tissue any further. So these two options are the correct answer for this question. We don't want these two things. So tissue engineering is basically not done for these two things. Rest of the options are correct. Making sense? Yeah, the explanation is in the next slide. You people can go through this once I'll be uploading the video as well as the PPT. I think that's all I have for you for this assignment, I guess. Yeah. If anybody has any doubt or wants me to repeat some of these questions in case you have missed it, I can go through that. If not, then we can stop the session here, and I'll meet you people next week, next Monday. Any doubts?
Ma'am, I have a query.
Yeah.
What does the term implant actually means? Is it the drug associated with the polymer?
No. So implant is basically, let's say, it could be an artificial organ, or in case of bone implants where you have, let's say, a titanium rod, that could be called as an implant. So, yeah, that's basically it. So the drug associated with that is that's not what we're talking about. So implant associated, the question is coming from the implant associated infections, right?
Yeah.
Yeah, so in that case you need to have a proper surface for the bacteria, for the infection to occur. So this concept is more relevant to tissue engineering instead of drug delivery. So in drug delivery also we were talking about these implants, right? The matrix and the reservoir system. That's not what I mean by implant here. This is more in case of tissue engineering. It could be an artificial organ or any other artificial uh, implant that you have put into the patient's body. Is it clear?
Yeah, what exactly does it mean in drug delivery? Because that's where I had the confusion.
What exactly does it mean in drug delivery?
Yes.
Yeah, so that's what I'm saying. It's not relevant to drug delivery as such. It's more relevant to the tissue engineering part. Yeah, so implant has no such meaning in drug delivery. So to let's say to avoid implant associated infections, you can use some of the principles of drug delivery. Like for example, coating the your particular implant. So by implant here, I mean any artificial organ which is not native to the body, or it could be the ceramic materials you can use. Let's say you have a root canal filling that is done, a root canal treatment. So you put a crown over it, right? For your root, after your root canal treatment is done, that can be considered as an implant. That is one of the examples.
Okay.
Yeah. Thank you.
Thank you. Thank you.
Anybody else? Is it possible to overcome implant association with suitable pre-adsorption of proteins on our implants, which might attract host cells? Yeah, you can do that as well. So there are proteins which are particularly, let's say, some of the growth factors which your cells need growth factors to proliferate and differentiate, right? So you can coat your implant surfaces with these proteins, which will then attract more of your host cells, the mammalian cells, instead of bacterial cells. That is also one of the methods to reduce implant associated infections. Any other doubts? If not, then I'll stop the meeting here. Welcome. Others, you have to discuss anything, or okay? Okay, see you people next week then. Thank you for joining.